Okay, students, so today the course code is CAM 445-1 and this is lecture 5A and the topic is antibacterial drugs and mechanism of action. And I am Dr. Musaddiq Ibrahim, Assistant Professor, Department of Chemistry, CUST. So, before moving towards the lecture, the objectives of this lecture is to understand that what type of drugs are used for antibacterial infections, and one of the objective of this lecture is to understand that by which mechanism or what is the mode of action of these antibacterial drugs by which they either stop the growth of the bacteria or they kill the bacteria. So, in this lecture series, we are going to understand that first of all, what are the different types of drugs, how they work, what is the stages of development during the whole course of time, and then later on in other lectures, we will go into the details about the mechanism of action of these drugs. So, about the history and development of antibacterial drugs. So, the discovery of antibacterial drugs starts from 1909 when Paul Ehrlich found the first pharmaceutical effective ingredient or substance or drug for the treatment of syphilis. And that drug or compound was known as salvarsan. So, this is what was the start of the antibacterial drugs to be used for treating different types of diseases or infections. Later on, arsphenamine was also used for treating infection, but it was found that it was highly toxic, which means that the curing effects were very less of this drug in comparison to the side effects or the toxic effects of this drug. Then later on, sulfonamide was the first sulfur drug, and it is actually in vitro derivative of prontosil dye. And this sulfonamide drug is active against streptococcal infections. So most of the streptococcal infections they occur in the throat or in the chest region. Sometimes they are also connected to the urinary tract infections. And because of the discovery of this drug, the Bayer's lab they were given the Nobel Prize in Medicine in 1939 because it was a collective effort. So that's why the company and its lab that was given the Nobel Prize for such a landmark achievement. Now, about the discovery of antibiotics. So, first of all, a penicillium mode was identified that produced a bactericidal substance and which was very effective against a wide variety of gram positive microbes or gram positive bacteria. So, this was the start of the discovery of antibiotics, and it came to known that actually what the substance in the penicillium mode is doing is stopping the synthesis of the bacterial cell wall. So that was the mode of action of penicillin that was causing the stoppage of the growth of bacteria. So as this penicillin, it was found that it is very effective, and during the times of World War II, there was no antibiotic available to treat the soldiers which were suffering from different types of diseases and infections. So at that time, when this antibiotic was presented, so it was found very effective and it was used widely during the World War II, and a very large scale production was carried out during those times. Later on, in 1943, the streptomycin was isolated from a cyanobacterium, which is known as Streptomyces griseus, by Salman Waxman. So, this was another antibiotic which was isolated in 1943 from a bacterium, and it was found that this antibiotic is bacteriostatic. So, the penicillin was found to be bactericidal, which means that it was killing the bacteria, while streptomycin was found to be bacteriostatic, which meant that it stopped the growth of the bacteria. And the mechanism of action of streptomycin was by inhibiting the process of protein synthesis by binding to the ribosome, which is the site where the process of protein synthesis is taking place and initiated and then executed. So, now moving towards development of antimicrobial drugs. So, development of new generation of drugs. So, around in the 1960s, new generation of drugs were produced. So, in 1960, scientists alteration or modification of the drug structure gave them new properties. So, actually, there was a lot going on in the 1960s that the already found drug structures were modified, and then they were assessed for their antibacterial properties, and it was found that some of the modified structure, they were more effective, they had more broad spectrum against different types of bacteria, including the gram positive bacteria as well as the gram negative bacteria, and that is how they were generating and developing different types of new antibiotics. So, penicillin G was altered to create ampicillin, and that's how the broadened spectrum of antimicrobial killing was achieved. So, the penicillin G, it was not a broad spectrum antibiotic, but ampicillin, which is a modified form of penicillin G. So the ampicillin became a broad spectrum antibiotic, and it was able to kill a broad range of bacteria. Now, moving towards the tree diagram showing the development of drugs with the passage of time. So, this is the tree diagram, and you can see that at the bottom in the center, it's the Penicillium chrysogenum, which is the mold from which a specific substance like penicillin G was produced. And then, so this is around 1940 and 50s. So then later on at around 1955, you can see that the tree in the middle it is written 6-APA, which is 6 amino penicillinic acid. And then on the right hand side, you can see that going from 1960 to 65, then 1970, then 1975, and up to 1980. So there is a huge development of new drugs by modifying the basic structure of the penicillin G or V. Some of the examples you can see on the right hand side during the 1960s are methicillin, nafcillin, oxacillin, cloxacillin, diclofloxacillin, and during the 1980s at the top you can see that you have temocillin, and on the left hand side you have different derivatives of penicillin like during the 1965s carbanicillin, ticarcillin. So these are different types of derivatives of penicillin which were developed by modifying the structure of the penicillin and then observing or researching the effects of the change structure on the treatment of the patient, and seeing the effects, observing the effects, that whether the modified drug is treating the infection or not. So it was found that with the development of new drugs, which were the modified form of the penicillin, so the new drugs they were effective as well as they were becoming broad spectrum antibiotic, which meant that they were able to kill different varieties, different classes, different types of bacteria. Now, moving towards mechanism of action of antibacterial drugs. So this is the important part, that how the antibacterial drugs are acting on the bacteria and either stopping their growth or killing them, and that's how the infection is cured, the treatment becomes successful. So normally when a person has a throat infection or a chest infection or intestinal infection, so that is because of the bacteria which is causing that infection. And then the patient is given that specific antibiotic which can then kill the bacteria, and as a result the patient recovers from the disease. So moving towards the mechanism of action, so some of the antibiotics they inhibit the cell wall synthesis. So the cell wall of the bacteria that is not being synthesized or it is being damaged when a certain specific antibiotic is given to the patient. So the examples of inhibitors of cell wall synthesis are penicillins, cephalosporins, vancomycin, and bacitracin. So these all drugs they have slightly different structure, but their mode of action is to stop the synthesis of bacterial cell wall. So, another type of antibiotics are those whose mode of action is to stop the protein synthesis in bacteria. And these antibiotics include aminoglycosides, tetracyclines, macrolides, chloramphenicol, and lincomycides. So these are actually stopping the protein synthesis of the bacteria. Then another one are those antibiotics which inhibit the DNA and RNA synthesis, that is nucleic acid synthesis. And the examples of these antibiotics are fluoroquinolones, rifamycins, to name few. Now, inhibition of metabolic pathways is another way by which some of the antibiotics they work, because likewise in human beings the bacteria they also have metabolic pathways for the synthesis, for the growth, for the reproduction purposes, and those metabolic pathways uh, are involving different types of enzymes, substrates, metabolites. So, at specific steps during those metabolic pathways, certain drugs have the ability to stop a certain reaction during a metabolic pathway. And the examples of those drugs are sulfonamides and trimethoprim. So, these drugs they stop the metabolic pathway, and as a result, the end product is not formed, and as a result the whole metabolism of the bacteria is disturbed, and as a result they are unable to survive. Further medicines or some antibiotics include polymyxin, which interfere with the cell membrane integrity. So the the top, the penicillin, they are stopping the growth synthesis of the cell wall, while the polymyxin it is interacting with the cell membrane of the bacteria, and actually the integrity, the the cell membrane structure is intact, but as of the application of polymyxin, the cell membrane structure of the bacteria is no more intact, and slightly gets disintegrated and eventually damages. So that's how some of the antibiotics are also administered for stopping the growth of the bacteria. So these are the some of the drugs which I have mentioned, and their mode of action, that how they are attacking different parts of the bacteria and either stopping their growth or killing the bacteria. And as a result, both in case of stopping the growth of the bacteria or killing the bacteria, the bacterial infection is compromised and the patient recovers from the infection. Now, moving towards mechanism of action about the cell wall synthesis inhibitors. So remember that here the cell wall we are talking about is of the bacterial cell wall, and the inhibitors are the antibiotics which are acting on the cell wall of the bacteria and stopping the synthesis of the cell wall of the bacteria, and as a result the bacteria is no more in its active form, and eventually it collapses, damages, and destroyed. So the inhibition of cell wall synthesis is actually happening that the drugs, the antibiotics, they interfere with the synthesis of peptidoglycan. So the peptidoglycan are the uh, units, the building blocks through which the cell wall of the bacteria is being synthesized. So when there is interference with the peptidoglycan, which is a combination of peptide, protein part, and glycan is the carbohydrate part. So these drugs, which are cell wall inhibitors, so they have a very high therapeutic index. So high therapeutic index means that these drugs are required in a very low quantity, but still they are very effective, and that's why uh, a small amount of the drug being given to a patient has a very enormous effect, and that is how the therapeutic index is being calculated. So if a drug is being given in high quality or high amounts, and the effective dose or the effect of the drug is less than the toxic effect of the drug, so then it means that that drug has a low therapeutic index. So the antimicrobials or the antibiotics of this class include beta lactam drugs, because in the structure of these drugs they contain a ring which is called beta lactam ring, and that's why these drugs are called beta lactam drugs. And these includes penicillin and cephalosporin, as well as the vancomycin and bacitracin. Right. So moving towards the cell wall synthesis inhibitors, so the drugs, they vary in spectrum, which means that some of the drugs, they are more active against gram positive bacteria, and some of the drugs, they are more active against gram negative bacteria. And with the passage of time, it has been found that the resistance is being developed by the bacteria against these drugs through production of beta lactamase enzyme. So this beta lactamase enzyme, it attacks the beta lactam ring of the drug, which we will see in the coming slides. And as a result of damaging the beta lactam ring of the antibiotic, the antibiotic structure is no more in its native form, and as a result, it cannot perform its function of killing the bacteria. So, through the process of evolution, the enzyme beta lactamase is being developed, is being produced by the bacteria, and then that enzyme is damaging or destroying the structure of the antibiotic. And then, as a result, the antibiotic becomes no more effective. So, in order to overcome this resistance, what the scientists have done is that along with the penicillins, they have introduced a beta lactamase inhibitor. So, the function of the beta lactamase inhibitor is to stop the activity of the beta lactamase enzyme, which is being produced by the bacteria to destroy the structure of the drug. So, the example is augmentin, which consists of amoxicillin and clavulanic acid. So, this is how the scientists are developing drugs and overcoming the resistance which is being developed by the bacteria. Now moving towards the structure of penicillin and cephalosporin. So this is the structure of penicillin at the top and cephalosporin at the bottom. And the orange color highlighted ring is the beta lactam ring in both the cephalosporin and penicillin. And if you carefully see the structure of the penicillin, so you can see that it starts from an R group which has been highlighted blue, then there is a carbonyl carbon, then there is an NH group, and then the NH group is attached to the beta lactam ring, which is CH, two CH groups at the top, and then carbonyl carbon and nitrogen connected together. And then afterwards, there is a sulfur atom, and then to the sulfur atom, another carbon atom is attached, and to that carbon atom, two methyl substituents are attached. And if you go down, so then again there is a CH group to which a carboxylic group is attached. So this is the structure of penicillin, and if you look at the structure of cephalosporin at the bottom, so rest of the structure is the same, but after the beta lactam ring, you can see that the sulfur atom ring has six atoms, and it has also a carboxylic group at the bottom, and then there is an R group which is connected to the C, which is being highlighted as blue. So the difference between penicillin and cephalosporin is that the R groups in penicillin is 1, and the R group highlighted blue in cephalosporin are 2, one at the start of the structure and one at the end of the structure. And these R groups, they are responsible for different modified forms of penicillin. So this R group can be a substituent, any benzene group or methyl group or ethylene group or benzyl group, and as a result of attachment of those functional groups in place of this R group, a different form of modified form of penicillin like ampicillin, amoxicillin, penicillin V, penicillin G, different types of derivatives of penicillins are produced, and those different derivatives of penicillin, they have different mode of activity. Some are broad spectrum and some are narrow spectrum. Similarly, in case of cephalosporin, you can see that you have two highlighted R groups present in the structure of cephalosporin, and as a result of changing the R groups, different derivatives of cephalosporin can be produced, and then they have different activity against gram positive bacteria or gram negative bacteria, or sometimes against both the gram positive and gram negative bacteria. Right. So the diagrammatic representation of cell wall synthesis inhibiting drugs. So this diagram, it shows you the structure of the bacterial cell membrane, which is colored as green and black, and then you have the violet and light blue color units, peptidoglycan units, which are on top of the cell membrane, and they are constituting the cell wall of the bacteria. So the different drugs, which are actually the cell wall inhibiting drugs, like beta lactam drugs, vancomycin, bacitracin, so they are interfering and attacking the cell wall of the bacteria and eventually breaking it down so that the bacterial cell is no more intact form, and eventually it is being destroyed and it is no more viable to reproduce or to grow. So if you look first at the beta lactam drugs, So they interfere with the formation of the peptide side chains between adjacent strains of the peptidoglycan by inhibiting penicillin binding proteins. So in the coming lectures, we will be more going into the details that by which mechanism the peptide side chains which are connecting the peptidoglycan units. So these side chains are being uh, not allowed to be connecting the peptidoglycan units, and as they cannot connect the peptidoglycan units, the side chains, so the integrity of the cell wall is being compromised, and as a result the cell wall is being damaged. So in case of vancomycin, so it binds to the amino acid side chains of NAM molecules. So NAM is the abbreviation of N-acetyl muramic acid, interfering with the peptidoglycan synthesis. And in case of bacitracin, so it interferes with the transport of peptidoglycan precursors across the cytoplasmic membrane. So it means that it doesn't allow the building blocks to be transferred through the cell membrane towards the cell wall, so that wherever these building blocks are needed for the synthesis of cell wall, so this is kind of like the raw material availability. So when these building blocks of peptidoglycan, they cannot be transported across the membrane, so it means that the wear and tear of the cell wall is taking place, and eventually the bacteria cannot survive and they are destroyed. So this was about the cell wall inhibiting drugs, their mode of action, and the references are from Medical Microbiology and Immunology by Levinson, the Basic and Clinical Pharmacology by Kordzung, Lipids Pharmacology, and Cell Biology by Harper and Rowe. So these are the books which are consulted, and for further understanding and develop further explanation and elaboration, you can consult these books. And okay, thank you.